I'm going to talk today about the power of collaboration in the context of the Coronavirus Immunotherapeutics Consortium, or the CoVIC. CoVIC is headquartered at La Jolla Institute for Immunology. I'm the program manager for CoVIC, and Professor Erica Ullman-Sapphire is the director of the CoVIC. We are all waiting for immunity against SARS-CoV-2. And for vaccines, the goal is to elicit antibodies against the SARS-CoV-2 spike protein. And we're doing a pretty good job in the United States with vaccination. As of late April, we had over 200 million doses administered. However, many people still are not vaccinated. Some are hesitant to take the vaccine. Some cannot be vaccinated. And for some, the vaccine simply will not work. For those individuals who become infected with SARS-CoV-2, antibodies can represent a potential therapy. Of the quintillion antibodies that are in human sera, multiple companies are looking to find the best antibodies against SARS-CoV-2, and they're using different strategies, finding different candidates. So the question is, which ones are the best? And are they better alone? Are they better in combination? And how do we make them affordable and durable and potent, and most importantly, accessible to many people across the globe? In March 2020, the Bill and Melinda Gates Foundation, in partnership with the COVID-19 Therapeutics Accelerator, reached out to Professor Sapphire to ask her to start the Coronavirus Immunotherapeutic Consortium, the CoVIC. The CoVIC is a global collaboration to look at antibodies from multiple discovery efforts to find out which ones are the best and what features make them the best, and how can we manufacture them economically so they can be available to the most people. This idea came from Ebola virus, which is what the Sapphire Lab works on in normal times. In 2013, an outbreak of Ebola virus disease started and went on to become the largest ever and claimed 11,000 lives. At the time, we had no vaccine, no drugs, and no treatments. Looking at this electron micrograph of an Ebola virus, and we zoom in, we can see that the surface is studded with proteins. These are glycoproteins, and they are the main target of antibodies against Ebola virus. The Sapphire Lab solved the structure of the Ebola virus glycoprotein in complex with a neutralizing antibody, and they also solved structures for other filoviruses that cause disease. So we understood from a structural basis how mechanical antibodies interact with the glycoproteins, and we knew that these all neutralized. But we had an issue. In 2013, we had antibodies that neutralized. This one here is a structure of KZ52, the Fab domain shown in orange, in complex with the Ebola virus glycoprotein. This antibody worked great in a test tube, but it did not protect non-human primates. On the right-hand side, we had an early triple cocktail. Of these three antibodies, two of them did not neutralize, but this cocktail fully protected non-human primates. So this raised a question. Did we need a cocktail for an effective therapeutic? Did it have to have an Fc function? And was the neutralization assays that we were looking at, were they right to predict whether an antibody was protective? So in the Ebola virus field, typically we'd have labs make a panel of antibodies and they would test their activity in vitro neutralization assays. That was the first criteria of selection and thought to be the best predictor of in vivo protection. Those antibodies that neutralized well were tested in mice, and those that worked in mice were tested in larger animal models of disease. So what we were doing was selecting for mechanical neutralization, but it was more difficult to look at Fc effector function. And so we needed to understand what were the features that were associated with protection. And to do so, we needed a statistically significant pool or a larger pool of antibodies to examine. So again, in 2013, we had antibodies that worked in vitro but didn't protect, and antibodies that protected but didn't neutralize. So we were missing information. We needed better tools. We needed more samples. We needed people to stop working in a silo, especially in the face of an escalating epidemic. We needed to work faster, to work smarter, and to work together. But the question was, can collaboration and competition coexist? So collaboration would give us a broader set of samples, a broader base of expertise, and allow us to perform side-by-side, apples-to-apples comparisons, and allow us, with competition thrown in, to move faster. Now, to protect people's ability to work on their own antibodies, we decided to blind samples that protected intellectual property. We also took parallel experimental tracks. One was a fast track to get us closer to a therapeutic, and then a comprehensive track to better understand what were the features of antibodies that best correlated with protection. And this resulted in the VIC, the Viral Immunotherapeutic Consortium, which is a global collaboration that involved 44 labs across 5 continents. We had labs from academia, industry, and government, all looking to see which antibodies are the best and what were the best ways to find them. This collaboration produced this data set looking at over 200 antibodies and more than 35 different features and 200 researchers working together. Christian Anderson's group at Scripps Research took those data, analyzed them, and came up with this network looking at how these different features correlated with protection. And also looking at antibodies against Ebola virus, they performed a logistic regression that identified 17 different antibody features that together are predictive of protection. Now, mechanical neutralization we already knew about, but this study revealed some new features that also could point to the ability to protect against disease, and these were all associated with Fc and the ability to recruit immune function. The VIC also showed that collaboration is possible, as evidenced by almost 80 peer-reviewed papers that came out of the VIC, more than 10 doctoral degrees, updates to the research pipeline, and most importantly, information that formed the basis for 6 new therapies, including a triple cocktail from Regeneron that substantially increased the likelihood of survival for those infected with Ebola virus. So now, in 2020, just over a year ago, as SARS-CoV-2 was emerging, we were tapped to form the Coronavirus Immunotherapeutic Consortium. Again, a global collaboration like the VIC to perform side-by-side, apples-to-apples analyses to look at which antibodies against SARS-CoV-2 are the best, what make them the best, what combinations can we find, and we'd make this data publicly available to everyone. The goals of the CoVIC, the primary goal is to save lives, to find an effective therapeutic that can be developed for deployment to individuals in low and middle income countries. And then also fundamental research, taking this body of data generated by the CoVIC to understand the landscape of antibody activities, and all of these compiled in the CoVIC database. And from that, we can look at what features are associated with which epitopes and which features correlate with protection. And we can also look at which assays are most effective in terms of predicting in vivo activity. Back in March 2020, we had a pretty broad criteria for acceptance of antibodies into the CoVIC. They just had to demonstrate affinity for the SARS-CoV-2 and preferably some functional activity, and also a willingness to enter into a global partnership with the Gates for development of the antibodies for low and middle income countries. We've updated those criteria, particularly for receptor binding domain or RBD targeted antibodies, and that's because we had quite a few RBD antibodies already in the panel. And so we asked people if you had an RBD binder to demonstrate that it was active against variants of concern and had superior potency. We are still looking for antibodies that target the NTD and the S2 domain. the workflow of the CoVIC, antibodies all come to La Jolla Institute for Immunology first, and as the program manager, I'm responsible for assigning ID numbers that allow the samples to be blinded. I then distribute the antibodies to the different reference partner labs who look at antibody potency, antibody characteristics, and all of these data come back and are deposited in the CoVIC database that allows the data to be analyzed to down-select antibodies. And the antibodies that are selected in partnership with Just Bio, we would look at sequence optimization to make sure we can manufacture these antibodies in the most effective way. We have 306 antibodies so far in the CoVIC panel. This is at the end of April. And these antibodies have come from multinational corporations and biotechs that are large and those that are quite small. Some have just a dozen employees, academic labs, and nonprofit labs. And the great thing about the CoVIC is because all these antibodies are blinded, they are all analyzed on equal footing, regardless of where they came from. There are a battery of assays that are offered to contributors to the CoVIC, looking at affinity, epitope binning, 3 different neutralization assays, in vivo protection, structural analyses, and Fc-mediated activity. And again, all these data are going into the CoVIC database. Contributors may view them in an unblinded fashion and also in a blinded fashion to see how their antibodies stack up against others in the panel. Biochemical analyses are being performed at Duke by Georgia Tomaros and Dan Bettinger at Cartera, using the Octet and Cartera's LSA platform to look at the binding kinetics between SARS-CoV-2 spike protein and the different antibodies, the ability to block ACE2 binding, epitope binning, and all of these assays are using structural biology grade antigens that are produced by the Sapphire Lab. Again, epitope binning by the LSA platform at Cartera allows us to look at blocking and sandwiching pairs and to have a network of how these antibodies interact with each other. High-resolution structural analyses here at La Jolla Institute for Immunology by Erica Ullman-Sapphire's group using either cryo and electron microscopy or X-ray crystallography. 3 different neutralization assays, pseudovirus on a VSV backbone being performed at Nexalis, Alexander Brukrayev's lab at UTMB at Galveston, looking at authentic virus neutralization, and Ralph Barrick at the University of North Carolina is also performing authentic virus neutralization. At Nexalis, the pseudovirus neutralization involves Vero6 E6 cells and a luciferase-based readout to give neutralization curves and IC50 values, as well as 80 and 90 values. For authentic virus neutralization at UTMB, they're using an M neon green readout for their assay, whereas the Barrick group is using a nanoglow luciferase readout, again producing curves in triplicate and IC50, 80, and 90 values. In vivo efficacy assays are being performed in partnership with Texas Biomedical Institute, first starting with the mouse model of infection using mice that are transgenic for human ACE2 under the control of a K18 promoter. We're looking at 13 mice per antibody. 5 mice go all the way through to the end of the experiment to look at survival and body weight loss. And then we have 4 taken at 2 time points to look at viral load, pharmacokinetics, and tissue distribution. We're first using a prophylactic model, and we will move to therapeutic when we down-select candidates. We are also working on a hamster model of disease. Yoshi Kawaoka and Pete Halfman at University of Wisconsin are performing escape assays for the CoVIC. And what they're doing there is incubating increasing amounts of virus with increasing amounts of antibodies and adding them to Vero E6 TMPRSS2 cells and culturing them, and then harvesting the P2 virus, and then from plaques, amplifying the DNA for the spike protein and sequencing. And we've seen actually quite a few spike mutations, perhaps more than we expected, given a virus that has a proofreading function. And importantly, we're seeing mutations that are also existing in circulating viruses, deletion mutants around residue 69 and 140, and then point mutations at residues 417 and 484. Other assays that we're performing are focused on the Fc end of things, resistance to AD and triple knock-in mice. Galit Alter at Reagan Institute is using systems serology in a high-throughput format to look at the immune profile of antibodies against the spike protein in terms of how they bind to the FcR, what their glycosylation profiles are like, and the ability to induce phagocytosis, NK cell activation, and deposition of complement. And they're seeing a wide variety of Fc profiles. Here in this pie chart, each slice of the pie represents another one of the Fc assays that they're looking at. Some have quite a bit of Fc-mediated activity, and some have very little at all. Sujan Shrestha at the LJI has developed a unique mouse model. This is a triple knock-in mouse that is expressing humanase 2, FcRn, and TMPRSS2, and we think that this may be a better reflection of the pharmacokinetics of antibodies in mice. And again, all these data come back to the CoVIC database, which is being built by Bjorn Peters at LJI and his team to allow us to look at the landscape of the antibodies. Contributors have an account to the database. They can go in and look at unblinded data for their antibodies, and they can also see in a blinded fashion how their antibodies compare with others in the panel. They can also use any data generated by the CoVIC for IND filings for their antibody candidates. The CoVIC database in a blinded fashion is publicly available. The website is here at covic.lji.org, and if you click on this arm of the stylized coronavirus, that will take you to the database. This is a snapshot of how the database is looking right now. If you see highlighted in gold at the bottom, I've chosen one of the antibodies. This is CoVIC-9, and you can see the different features. So we have a table below and up above in tile format representing how this antibody stacks up. If we zoom in, we can see that in these charts, the antibody you've selected is highlighted in yellow. So on the upper left, we have how that antibody compares in terms of neutralization. This is a pretty potent neutralizer. Also, how it looks in terms of the epitope community, which we'll talk about in a minute, and where that epitope community is on a structure of the SARS-CoV-2 spike, which is shown here in red. And last, we have a partnership with Betty Korber at Los Alamos National Laboratory. She is tracking spike mutations and how they are being transmitted among humans. And this is important because there are emerging variants of concern. In late April, Eli Lilly asked the FDA to revoke its EUA for its COVID antibody therapeutic in order that they could focus more attention on a combination therapy. And this highlights the importance of choosing antibodies that will have durable resistance to multiple variants of concern. And to talk a little bit about some of the data that is coming out of the CoVIC, this is epitope binning of the first 200 antibodies in the CoVIC. This bin was done by Dan Bettinger on the LSA platform using soluble RBD as the antigen. And you can see that there's a variety of different communities. We have 11 shown here. And we've mapped these on the SARS-CoV-2 spike protein. This view is looking down on the molecule. And we can see some shown in orange and yellow target the receptor binding motif, some on the outside face of the RBD, the inside face on the up with RBD in the up conformation, and then some that target the down conformation, and then the N-terminal domain binders NTD 1 through 3. We have structures, negative stain structures, for representatives of each of these communities. Here the SARS-CoV-2 spike protein is shown in gray, and the Fab domain is colored. And you can see the variety of binding positions looking at the top view and the side view for each of these epitope communities. Looking at the neutralization in terms of epitope bin, we can see that RBD community 11, RBD 11, has the lowest IC50 value in terms of the mean. Antibodies that target the receptor binding motif are highly susceptible to the emerging variants that we tested. Meanwhile, those in community 6 that target the receptor binding domain coming from the outer edge, those are more resistant to emerging mutations. And community 11 are also resistant to emerging mutations. And what these data show is that we have a variety of antibodies to choose from, and we can develop partners between them, combinations that might together be resistant to these variants of concern. And we can see that the CoVIC is fostering partnerships. We have partnerships between contributors who are looking for combinations to flesh out their therapeutic candidates and to modify their antibodies to work in novel formats and to use the data in IND filings. The CoVIC is supported by the COVID-19 Therapeutics Accelerator, a partnership between Bill and Melinda Gates Foundation, the Wellcome Trust, and others, a supplement to the VIC from NIAID and the GHR Foundation. And I am delighted to be a part of the CoVIC and to work with the director, Erica Ullman Sapphire. And I'll be happy to take any questions.